Hey guys, so today we'll be going over how to synthesize purines. It's a little blurry. There we go. And we know we have two types of purines, adenine and guanine. So we have A and G. So the first substrate that we start with is actually the products of the pentose phosphate pathway. If you know that we make one of the products or ribose 5-phosphate as well as NADPH, but for right now, let's just talk about ribose 5-phosphate. So we have ribose 5-phosphate. This was just from the pentose phosphate pathway. So you know it's a ribose, typical ribose, with on the 5th carbon there's a phosphate right here. So what actually happens is that we take this substrate and it undergoes an enzyme, or it interacts with an enzyme, which is called PRPP synthase. So PRPP synthase. So what it actually does is that it takes ribose 5-phosphate and it makes PRPP from it. So what is PRPP? Well, it stands for phospho ribosyl pyrophosphate. So let's look at these components individually. We just draw this out. So this was our phosphoribosol. So this is our phospho right here. Ribosol is our sugar, and pyrophosphate. If you remember back to just biochemistry, pyrophosphates are just two phosphates with one another. So on the 1st carbon, we'll just be linked with 2 phosphates. So this is the first reaction that occurs. Next, what actually happens is that this molecule right here is called PRPP, phosphoribosyl pyrophosphate. This then interacts with an enzyme, which is PRPP, same thing, amidotransferase. And in a sense, let's just look at what the enzymes are named so we can get a value for what their function is. Amido, meaning amino group, is going to be transferred onto this PRPP molecule. And it's going to be right on the pyrophosphate groups, so on the 1st carbon. So now we will have a molecule that looks like this. We have this amine group right here. And this is going to be called 5-phosphoribosylamine. Now we have the amine group right here. Did I spell this right? Ribosyl. So there's sugar right here. And on the 5th carbon, right here, we have a phosphate group. Next, the other steps aren't that important, but it's important to know just a couple things. These enzymes are most important, this PRPP synthase and this PRPP amidotransferase. What happens is now that it's going to interact with glycine, aspartate, glycine, aspartate, glutamine. It's also going to get THF, which is going to be a carbon donor. But that's not really that important. All you have to know is that it will turn into what's called an inosine monophosphate. So it'll look something like this. And in a sense, this whole purine synthesis, all we're doing is really changing this 1st carbon, what's actually in it. Now after this, we have what's called hypoxanthine. And we still have this 5' phosphate group right here. This is what I was referring to as inosine monophosphate, or IMP. So this is actually a nucleotide. You know, it has a phosphate group, a sugar, something on the 1st prime carbon. But how do we turn this into either adenine or guanine? Well, it can either undergo 2 steps. It can either get an amino from glutamine. So if you get let's write NH2 from glutamine, or it can get an NH2 or an amino again from aspartate. If it gets from aspartate, it turns into adenosine monophosphate. If it gets it from glutamine, it turns into glutamine monophosphate. So this is how we essentially make these two purines. And these purines are the two cyclic heterostructures. One last thing that you should know is how this IMP, this GMP and AMP can actually come back and negatively inhibit aminotransferase, because the cell only makes what it needs. It's not going to make any more, so there is negative feedback inhibition. And another thing that you should know is this whole process is taking place in the cytosol of the cell. Cytosol of the cell.